more importantly, we had computers that could finally do something useful with respect to this goal. They could first of all store the images, could look at them on a monitor. And then over the past 20 years, we built increasingly sophisticated machine learning systems that can analyze the images and trace the neurons and find the synapses. Look, ultimately the goal is to understand how the brain works. That's why people fund this type of work. That's really the first order of business. And, more specifically, the reason people want to understand how the brain works is to improve human health.
Hi, I'm Craig Smith, and this is Eye on AI. The structure and function of the brain has long inspired artificial intelligence research, while AI has helped neuroscientists understand the brain. In this episode, we glimpse into the painstaking efforts to reverse engineer thought by mapping the brain's neural pathways, a process called connectomics. Viren Jain, who leads Google's Connectomics team, discusses the history of the science from early microscopic investigations to modern large-scale electron microscopy datasets aided by AI. Viren shares his perspectives on how connectomics could inform our understanding of learning and intelligence, and talks about Google's participation in a National Institutes of Health initiative to map 10 to 15 cubic millimeters of mouse brain over the next 5 years. I hope you find the conversation as fascinating as I did. Hi, this episode is sponsored by Celonis, the global leader in process mining. AI has landed, and enterprises are adapting, giving customers slick experiences and the technology to deliver. The road feels long, but you're closer than you think. You see, your business processes run through many systems, creating data at every step. Celonis reconstructs this data to generate process intelligence, a common business language. With process intelligence, AI knows how your business flows across every department, every system, and every process. With AI solutions powered by Celonis, enterprises get faster, more accurate insights, a new level of automation, and a step change in productivity, performance, and customer satisfaction. Process intelligence is the missing piece in the AI-enabled tech stack. Search Celonis, Celonis, to find out more.
So yeah, I'm Viren. I'm a research scientist at Google. I lead the Connectomics team there. In terms of my background, I got a PhD at MIT, working sort of at the intersection of computer science and neuroscience with Sebastian Sung. When I first started graduate school, we were working on sort of more theoretical ideas, sort of how would you mathematically think about how brains work. But we sort of realized that we didn't really have the data we needed to think about that or to advance that in quite the way we wanted. So then we turned to this other enterprise of really just trying to actually map out brains in detail and sort of from the bottom up build the data that we needed. So after graduate school, I went to a place called HHMI Janelia. So the Howard Hughes Medical Institute has an institute devoted to these kinds of topics. And I ran a lab there for a few years. And then in 2013, I moved to Google, where I started this Connectomics team, and for the past 10 years have been working on and developing the effort associated with that here at Google Research.
There's been a lot of work done on single cell, and there's one particular worm that they've pretty much mapped. Can
C.
you
elegans, yeah.
can you give us that history before we get to Jeff Lichtman and
Absolutely.
mice? Yeah.
Yeah, so the history of the field was, that goes back in some ways, even to the earliest days of neuroscience, where Cajal was, about 150 years ago, looking through microscopes and staining neurons in different organisms. What he noticed, and this is really a key insight in some ways, is that neurons have different shapes. These shapes are interesting because they support the general organizational principle of biological brains, which is that you have these units called neurons, and they, form connections with other neurons And the hypothesis was that there was a lot of information, a lot of important functional information encoded in how the neurons actually talk to each other. Which neurons form connections, which with each other ones, and so on. And so this idea kind of started to take root in neuroscience a long time ago. And then in the 70s, Sidney Brenner, who was a scientist at the MRC in London, at the Laboratory for Molecular Biology, he decided to pursue what was at that time a very novel idea, which was what if we just mapped out all of the connections in a small organism. And he chose this organism called C. elegans, which is
Right.
a roundworm with 302 neurons, had already become popular in genomics research, and was beginning to become more popular for other forms of biological research. And so they took one of these worms, they sliced it into 1,000 sections. They sort of imaged each of those sections with an electron microscope. And then interestingly, they printed them out. So they took all these sections, these basically literal films. They printed them out onto different sheets of paper, and then for about 10 years, or maybe 5 to 10 years, they sat around tracing the neurons from one section to another to figure out where all the wires are going and who they're talking to. And from that, they were able to reverse engineer this so-called connectome, describing the chemical synaptic connections within that worm brain. So that effort has actually been quite influential. People who study worm brains use that information all the time to guide their experiments. But it was such a pain, right? 10 years essentially for 302 neurons, that people, became discouraged from trying to repeat the feat, for anything larger than a worm brain. Until maybe, about 20 years ago, around 2004, uh, people kind of revived this idea and thought, now, we can do two things. First of all, we can automate the process of sectioning and imaging tissue in this way. We can build automated platforms that will slice up tissue and image them. But more importantly, we had computers that could finally do something useful with respect to this goal. They could first of all store the images. We could
Mhm.
look at them on a monitor, and then over the past 20 years, we built increasingly sophisticated machine learning systems that can analyze the images and trace the neurons and find the synapses. So, over the past 20 years, this field has really progressed, much more rapidly. There's been a lot of progress on mapping the entire fly brain. Which is some work that our group has participated in. There's been a small amount of work done on mouse and human brains. So you mentioned Jeff Lichtman earlier. We worked with his lab to reconstruct a cubic millimeter of human brain tissue, which was a very interesting project. On the one hand, one of the largest data sets ever collected in biology, 1,400 terabytes of data. But on the other hand, it was one millionth, so that's one over one with six zeros in front of it, 1 millionth of an entire human brain. The technical challenges in scaling this up are really quite daunting. And what the field has converged on is the idea that after the fly, the next big milestone could be the mouse brain, which
Mhm.
is about 1,000 times larger than a fly, but still 1,000 times smaller than a human brain, and is obviously a nervous system and general organism, which is studied commonly in biology.
And for the roundworm that involved this machine, is it the same machine that Jeff Lichtman uses? And was that then the same method for the fly brain that you're taking these? I don't know what is the thickness of the slices of the brain.
So, conceptually, the approaches are all similar in that, what you're doing is, using some combination of cutting tissue at very thin scales. So, anywhere between 10 to 50 nanometers, it's kind of like human hair thickness or smaller. Um, and then you're imaging the tissue using some kind of electron microscope.
Hmm.
Now, the details can get quite different though. And just to give you a flavor of this without getting too technical, there's broadly speaking two different methods in the field: something called serial section approaches and another one called block face approaches. And in the serial section approach, so this is really like the deli slicer idea, where you take a brain, you slice it up into independent sections, and then you image each of those sections independently. And you have to be very careful because those slices are very thin and delicate. But the advantage is, once you have the individual sections, you can image them with whatever kind of microscope, electron microscope you want. Um, the other approach in the field, which was used to do some of the work which I mentioned, is the block face approach. And just real quick, the idea is you have like a block of tissue, you bounce electrons off the surface, and then you ablate. You either shave off or literally vaporize the top layer so you can expose some more tissue to be imaged, and you keep alternating those two steps. Anyway, the point is it's a real technical challenge to image a piece of brain in 3D at nanometer resolution. And so there's been a lot of ingenuity put into, how can you do this process reliably at scale with higher throughput? And that continues to be a big debate in the field, just like in genomics, where, there was a 30-year, progression of technology that continually made sequencing genomes cheaper and more efficient. In many ways, we're trying to do the same thing here to try and invent, the raw data acquisition techniques, which could image brains at scale and more cheaply than in prior projects.
Yeah, and in once you have the images of the slices stained, you were saying these guys, I don't know if you meant literally printed out sheets of paper and were tracking neurons from page to page. That's all done with artificial intelligence now, isn't it?
That's right. So yeah, certainly we're not printing anything out anymore. Thank God. That would be a lot of paper for a fly brain even. Um, and so computers are used in many respects. So first of all, just to store the images and organize them and
Hmm.
and recapitulate them into a three-dimensional object. That's certainly one task that needs to be performed. Second is, as I mentioned, this process of tracing neurons. So the way to think about this is, you probably, have heard of like server closets, right? Where you
Hmm.
have like a stack of computers, and in the back there's a bunch of wires connecting them, right? Imagine taking a black and white photo of a server closet and then trying to ask, okay, what's the wiring diagram of, all of how the computers connect, right? The only way to do that is to, find a wire and visually trace it until you end up at some other machine, right? There's nothing, they're not color coded, there's nothing, there's not some number on the wire which tells you where it starts and where it goes to. And so the way to think about the brain when we image it with electron microscopy is like it's like a three-dimensional server closet. And so if we want to figure out the wiring diagram, we have to literally trace out, where one wire begins and where it ends. And that's quite a difficult task if you think about, each of these wires is, can sometimes be like tens of, or hundreds of nanometers thin, and they span, millimeters to centimeters of tissue, and they're all jumbled together in like this giant mess of spaghetti. So doing that process accurately, and at scale has been non-trivial to automate, but is something which, a number of us have put a lot of effort into designing machine learning algorithms for. That said, the algorithms are not perfect. So the final stage of all of this is actually to have humans go through and fix all the mistakes or fix all the things that that computers cannot do. And that remains sort of like maybe the biggest bottleneck in this whole process is that we still need thousands to tens of or hundreds of thousands of hours of human effort to finish a given nervous system. And one of the big practical and research challenges in the field for us is to continuously drive that number down. So the next time we do this, we need only 10% of the human effort that we did last time, and so on and so forth.
Yeah. And just on that point, I was just talking to somebody about reinforcement learning and with AI feedback and having multiple AI agents uh, uh, sort of debating with each other uh, to converge on a final answer, to try and combat hallucinations and that sort of thing. Are there strategies like that where if there are errors in the in the map in the connectome that's been traced through machine learning, that you could have adversarial algorithms that eventually figure out what the accurate path is?
There have been attempts to sort of combine the predictions from, some diverse set of algorithms to improve the overall performance of this tracing process. It has been difficult to do that in a really sort of meaningful way. Just it tends to be the case for this task that if one algorithm is wrong, many of the others will be wrong also. But there have been attempts to do that. For now, I would say the push has really been, once we've had some amount of human effort applied to the hard problems or the hard cases, can we go back and retrain, the initial algorithm with all of that additional data to have it be more accurate in the next instance?
Yeah. So with the mouse brain, I mean, how far again? What percentage of the overall brain have you completed, and how long did that take, and all the metrics surrounding that?
So the mouse brain project, that's really, that's just getting going. So that's, the announcement last week was really, a suite of projects that the NIH has funded to really scale up mouse brain connectomics. What's been done there so far has also been at, let's say, the cubic millimeter scale, which, if you think about a whole mouse brain as being 500 cubic millimeters, so that's 1/500th of a mouse brain that's sort of been achieved so far.
Hmm.
And the goal of these new projects that the NIH is funding, of which we're part of one of them, is maybe to do 15 cubic millimeters or so. So scale up maybe 15x and get let's say a few percent of the whole mouse brain at the end of the day. That said, that's sort of the goal for the next 5 years is to hit that milestone and see which technologies are scaling best and are producing high quality data. If that's successful, it's possible the government will fund a second 5 year effort, which will really try and do the whole thing. So to pick a particular technology, which seems to be going well, and then put in the money and resources to do the complete map of the mouse brain based on that.
And what do you learn from the connectome? That's one question. Um, because this is looking purely at the physical structure, not at the
That's right.
biochemistry or any of that. And then, uh, well, why don't you answer that first?
Yeah. Well, there's a real sense in which we're still learning what we learn from the connectome. But we've had, I would say, a number of interesting examples so far. So first of all, in C. elegans, where we've had the connectome for a long time, that data structure, so to speak, having that information in C. elegans, has been transformative for studying that nervous system. So people could, for example, go in and ask, okay, what are the neurons that are connected to the motor output of the system or the sensory input? Or what are the ones which, are just sort of in the middle of all the processing? And, if you want to then study those particular aspects of the nervous system, you know where to begin. Or if you want to ask questions like, how does the information flow accomplish some objective, some behavior that the nervous system has. That connectome will give you a number of different clues and hypotheses, which you can then use to decide your experimental strategy. Similarly, I would say the fly brain, which is a much more recent story, the connectome has really been a big success there, I would claim, in the sense that almost anybody who works on studying the fly nervous system, of which there might be more people than you imagine, basically now uses again that data structure in all kinds of ways to guide their experimental strategy. So you can sort of trace the information flow throughout the brain and say, okay, well, it comes into these receptors and then it goes to this part of the brain that processes, let's say, smell information, and then it goes to the part of the brain that stores memories, and then it goes to a part of the brain that decides what action the fly should make. And we've really gotten to the point in the fly brain where for some specific behaviors, you can literally trace out the synaptic steps that are involved in computing some piece of information or making a decision, and start to really build a detailed model and hypothesis for how the brain works. So I would say, in the worm and the fly, there's now very good support for the idea that having the connectome can be extremely valuable for understanding how the system works. I think as we scale up to a mouse and one day maybe a human brain, um, things will get a little bit more confusing, right? So I mean, the difference, one of the main differences between a worm and a fly and us is that much of our brains are there to achieve learning and memory and experience
Hmm.
driven changes in behavior, right? A worm is largely born with the behaviors it's destined to accomplish. A fly, mostly, although it does do a little bit of learning. What's interesting about certainly human brains is we do a ton of learning, right?
Hmm.
Much of our day-to-day behaviors are driven by things we learned after we were born. So in that sense, how interpretable the connectome will be in some of these larger systems, and what form these explanations will take of neural circuits, it's a little bit less clear. But in some sense, we're excited to find out.
And are there any sort of principles from now that you've got uh, the roundworm? I'm not going to try and pronounce the name of the
Sure.
roundworm, and the fly. Uh, that you can see principles that apply to each, maybe in the shape of neurons carrying certain kinds of information and that sort of thing.
Yeah, absolutely. I mean, so one of the most Uh, one of the most striking things in both, I would say, the worm and the fly is how closely related we found structure and function to be. Where the particular shapes of the neurons and the connectivity that we observe just in the connectome um, really does seem to, excuse me, have a close relationship with the function of those neural circuits that we observe when we go in and measure their activity and how they influence behavior, and so on. So there are parts of mammalian nervous systems in which we see a similarly strong relationship between structure and function. So, for example, in our retina, which is the part of the brain that's at the back of the eye and
Hmm.
first processes light, we see again a very strong relationship between sort of the shapes of neurons and their synaptic connectivity and what their functional role is. So, I think that is one principle which really has quite a lot of power and has been proven to be true in some of these smaller organisms.
Hmm.
Now, I think as we move to some of the higher processing centers, so to speak, in mammalian systems like the cortex, um, there it seems a little bit more like a computer, to a first approximation, where you have a bunch of sort of generic parts that get wired together in all different ways, and the relationship between structure and function there may not be quite as obvious. And so I think those will be interesting cases in which the analysis that we do of the connectome might have to be much more subtle and complicated in order to figure out what's going on.
Yeah. You can see that uh, shape and function in these lower level brains, or is that also applied to the lower brain in the human? But you can see a connection between shape and function. Are they? Is there some sort of a commonality between this kind of shape in the roundworm is analogous to this kind of shape in the fly, and they're performing similar functions? I mean, can are there any is there any deduction that you can
Right,
make
right.
from that? Yeah.
So I would say the general concept which you're getting at is what we call cell type, right?
Hmm.
So can we cluster all of the neurons that we see in one brain or across brains into categories, right? And say this is one type of neuron versus another type of neuron. And based on doing that, can we then infer, other properties of them, like what their functional role will be in a circuit and so on. And this is a very active area of investigation for neuroscientists. So this isn't just about connectomes. So there are people who are, measuring gene expression to see, which genetic
Mhm.
programs are active in different brain cells and using that to categorize cell types, and so on. But in general, yes. I mean, the answer is definitely that, there seem to be, some discrete set of types of cells, which, have different genetic specification and different shapes, and different physiology to go along with that, and different roles in the nervous system. I think going from the worm to the fly, that's a pretty big evolutionary jump,
Mhm.
actually. And it's not so obvious that the types we find in the fly are really that closely related, to the types that we find in the worm. The worm is kind of an interesting case, right? Because they've been around a long time, right?
Mhm.
Hundreds of millions of years at least. And they're small. They're 300 neurons, so they've just been hyper optimized by evolution, to do what they do efficiently. And it's almost kind of this like boutique analog circuit where every part in the system is a little bit different, and playing multiple roles at the same time, and so on. And this is very different than if you go to, let's say, a human brain, right, which has 100 billion neurons. There's no possible way the genome could encode details of 100 billion different neurons. So what you have is a much more modular organization, right? You have a small number of cell types, maybe thousands, which get repeated into different motifs and organized in different ways. And so, the sort of scheme of the nervous system there is very different.
Mhm.
Um, so the fly is then kind of like in the intermediate, where there are definitely some very specific cell types. It's maybe not all genetically encoded, but quite a lot of it is. We see a little bit of learning and memory, so we're kind of getting a taste of both styles of computation there. But coming back to your question, the identification and characterization of cell types is one of the most sort of pressing tasks in neuroscience, because that's really what will let us simplify in some ways our models of these systems. If we have to think of each neuron as sort of its own special thing, it's going to be impossible to really develop a description that's interpretable.
The other thing is from what I understand, again, I'm just a journalist. Uh, neurons, the connections of neurons in the brain can change over time, and even the shapes can change over time, if I'm not mistaken. I mean, so when you're slicing uh, a brain, you're sort of capturing a moment in time. Is how do you track the changes in neural patterns?
That's right. So, I mean, definitely with connectomic methods, we're looking at a static point in
Mhm.
time for that particular organism. The method is, highly fatal, I would say. Your brain ends up as a series of slices. So, there's no more activity or changes. Um, so it depends on the organism that you're studying, right? For worms or flies, um, there certainly are dynamic elements there, but they appear to be maybe less critical to the overall story as compared to a human brain, where again you can learn entirely new behaviors within a short time span, and obviously encode new memories, and so on. So I think studying static connectomes will let us make progress on quite a lot of aspects of the nervous system. Even in human brains, it's not that things are getting completely reorganized or even mostly reorganized from one day to the next. In fact, most of the structure is very likely to be quite stable over long periods of time, because we have to support all of this stable physiology, and all of the sort of stable behaviors that we have over many decades. So I think the connectome will be extremely useful in analyzing those types of issues in neuroscience. I think specifically for learning and memory, it's a bit more challenging for these methods. But certainly, it's not impossible. People have, been looking at ways to, let's say you take two different flies that have had two different sets of experience, and you look at differences in their brain. Can you pinpoint that, using methods from connectomics? But I should say, neuroscience has many different experimental tools available to it. There are people who, just image the activity in the brain or just look at specific connections over time. And really, it will be the combination of all of these different, measurements and experimental tools, which will be required to explain, really the most complex issues in the brain, like, how do we encode a memory or how do we learn things?
Yeah. When you're doing this, you're taking cross sections at some point of neurons. You're obviously not able to, uh, slice so that you have one layer of whole neurons and then another layer of whole neurons. How much can you see into the internal structure of neurons and axons and dendrites and all of that?
Yeah, that's an excellent question, actually, and quite a bit. So, what you have with an electron microscope is really a very high resolution, unbiased instrument for probing the structure of biological tissue. So, while with connectomics, people typically talk about, okay, well, what are the list of connections between cells? But really, you have much more detail that you're getting out of this data set. So, first of all, you have the precise shape of each cell, which can often be very interesting. And then, as you mentioned, within each cell, we can often see, all of the organelles. So, all the mitochondria, the nucleus, microtubules, all kinds of different internal structures which support the physiology of the cell. And an increasingly interesting, goal in our field is to understand how are those details related to, what the brain is doing overall. So if a neuron has one function versus another, does that imply that its internal organization, the kinds of organelles it has, and where they're situated is different? Another very, important structure are the synapses, which are, the actual sites
Hmm.
of communication between two neurons. And, we can often in our pictures, we can see them and we can see the individual vesicles, and we can see their shape and size, which is correlated with various aspects of their physiology. So characterizing, all of that detail in the images is a big job, and something which, again, we're applying a lot of machine learning and AI towards automating, so that in our descriptions of these circuits, we don't just have this super abstract A connects to B, but we have a lot of information about A and a lot of information about B, and what their internal structures might be like.
Hmm. Yeah, it's just making me think when I was doing some reading about Jeff Lichtman's work. I don't know if it's his lab, but they produce these beautiful images of stained uh, cross sections of brain that you can buy and hang as wall art. Uh, is that the resolution that you're working on, or are you working at a greater resolution than what I'm referring to?
So I guess I'm not exactly sure, which images you might be referring to. I mean, I can say that, the data we work with, the resolution is measured in the nanometers, so
Mhm.
anywhere from 5 to 10 nanometers for each pixel. And so, that's, you can always go higher. So, if you want to see the internal structure of an individual protein, then you have to go to, single nanometer or higher resolution. But if you want to see how a cell is organized, that's actually, you can see quite a bit, at a few nanometer resolution. And that is what our entire data sets comprise. And, the reason they're so high resolution is that, the machinery that the brain uses to establish these connections, the synapses and the individual axons, they often become very, very small,
Hmm.
at that scale of nanometers. So if we want to actually resolve all these connections, it turns out we need to both image at very high resolution in order to see those connections, but also over very large fields of view, in order to see where they go. And from a technical point of view, that's really what makes this field very challenging.
Mhm.
We have lots of methods which can image large brains if that's what you want to do, and we have lots of methods that can image at very high resolution. But here we have to do both at the same time in order to make sense of things.
And does your work involve uh, you're doing the mapping, but are you also uh, making hypotheses about the function of different shapes or different uh, structures that you're seeing within the or the different connections uh, for how not only how information flows, but what kind of information flows through different neurons. I mean, for example, visual information or um, some other sensory information.
Right. So, um, in terms of the field as a whole, those are definitely critical topics which people study using this data. At Google Research, we've chosen to specialize on the parts of the process where we can sort of add the most value. And that's really, we don't collect the raw data. We work with people like Jeff or HHMI or Max Planck, so they're collecting these huge data sets. And then, we sort of sit in the middle. We sort of take that data, we turn it into a form that's, annotated and traced, and then is usable by biologists downstream who might be interested in all sorts of questions. So, we don't really see our role as, necessarily, solving the biological questions or inferences, but really as just taking the raw data, applying a ton of computational tools and techniques and machine learning algorithms to turn it into a form where then a biologist, who is interested in some particular question, could use it. And so, to do that, we've developed ML methods to solve that tracing problem I mentioned, or as you were just referring to solve this problem of cell types. If I just give you a small fragment of a cell, can I tell you, what type of cell that is? Because then you'll know a lot more about what to expect its function to be, and that's a problem which we've worked on and have a paper coming on about in a few weeks.
Yeah,
So that's really where we see as the area which we try to focus on.
and what is Google's interest in this? Because is it is this a service that you're providing to the research community, or are you building product? Whether it be data sets or techniques, algorithms that then can be applied elsewhere. Because it,
Yeah, yeah, no,
yeah,
absolutely. I mean, this is primarily a research endeavor, so we're not charging our collaborators. In fact, with this new project with the NIH, we are working with a bunch of people who they're getting money from the government, but we are not. We are contributing our work and our resources, pro bono towards this goal. So, I think there's a couple of motivations here. I mean, one is to see how computer science can actually have useful impact on some of the fundamental problems in the natural sciences. And this was one area where there was these huge data sets and these machine learning problems, and it just seemed like a good fit for the kinds of things which Google has historically excelled at. You think about mapping the web, mapping the earth. Why not jump in and help map the brain? That said, there have been things that our team and collaborators have achieved which have been useful in the company more broadly. So, for example, one of the most basic things we had to build was infrastructure to store and manage all of this huge data, right? So, we get literally millions of images, which then need to be, recapitulated into a three-dimensional coordinate system. We need to annotate them, visualize them, process them. To do this, we built a system called TensorStore, and it turns out that system, has been useful for other teams at Google. So, for example, the teams that are training these huge large language models or other machine learning systems ended up using that to store all the parameters of those models. So, there is this general research intuition that if you choose a really hard problem and make progress, you'll end up inventing things that are more broadly useful. And in computer science, that's especially likely because the problems end up being very general. So if you pick a really hard machine learning problem or data problem, you'll kind of have to invent things that are more broadly applicable.
Yeah, and then so that's how computer science or AI is aiding in this research or this discovery. But there's also does this kind of work give you any insight into how the brain learns? And the reason I'm asking is I talk uh, periodically to Jeff Hinton, whose primary goal is to understand how the brain works, not to build, you know, big profitable AI engines. And his frustration has been that back propagation, uh, after decades of research, is the conclusion is that that kind of an algorithm does not exist in the brain. And he more recently has work on something called the forward algorithm, which he thinks could possibly work in the brain, where information is passed back and forth, but not back along the same pathways. And is that kind of thing something that you could find evidence for, or are we no longer talking about the physical structures and more about well, you would be ultimately, because you're talking about electrical impulses and changing weights and from
Yeah, I
neuron to
mean,
neuron.
this is a great question, Greg. So, we have a project actually where we're looking at this. So, I'll tell you a little bit about the project and the species, the organism which we're studying in order to shed light on this is something called a zebra finch. So, it's a bird. And the interesting thing about these birds is that they learn their song from their tutor, typically their parents, but some other bird. When they're young, they're born not knowing a song, and then over a period of weeks, they learn a very specific song. And Neuroscientists have been very curious about this process because it's sort of a stripped down version of the learning problem, right? I mean, how do they
Mhm.
go from not knowing a song to knowing a very specific song in a matter of weeks? And it's interesting precisely for the reason you mentioned, because in general, neuroscientists do not think backpropagation is sort of a plausible explanation for how such a learning process might work. And so there have been a number of very specific theories put forth about how in this bird this might work. And behaviorally, it's interesting because the bird starts out babbling, right, making a song that's incorrect, and then they slowly and slowly get better, right, over a period of days to weeks. They keep tweaking the song, and each time it's a little bit more like the song that they're supposed to make. And so people have identified a bunch of circuitry in the songbird brain, which seems to be associated with this process. And what we've done now in a preprint and a project that is still ongoing is we mapped out the circuitry in one part of that part of the bird brain. And indeed, we find evidence for a very specific type of learning algorithm, something called node perturbation, which is an alternative to backpropagation. And this is basically a mathematical alternative to the type of gradient learning which backpropagation achieves, which was proposed 40 years ago. And we found some very specific types of connections and patterns of connections which would support this type of learning algorithm versus others. And this is just the beginning of this type of work. It's this is not easy, and like the inferences are very subtle at this stage. But we're sort of excited to take this further because, as you mentioned, I mean, this is one of the key problems in neuroscience, right? Brains seem to be doing all of this learning and adaptation, but if backpropagation is not the answer, what is?
Mhm.
And how does it do it efficiently? Probably, one of the most fascinating things about, biological intelligence, is, for example, how energy efficient it is.
Hmm.
Where, a human brain uses basically, a light bulb's worth of energy at any given moment, whereas, big machine learning systems can use much more than that. So, another big question which, we're interested in is, regardless of sort of, ultimate performance or accuracy, how is it that biological systems manage to do it with so much less energy and, so much less computation than the artificial systems we have?
And so much less data also. Yeah.
That's
Yeah.
right.
The I had Rich Sutton on the uh, podcast a couple of years ago. I mean, it made me think when you're talking about this Finch. Is that
Yeah,
the
zebra finch. Yeah.
learning this song, and it gradually gets better. It makes me think uh, of his temporal difference learning algorithm.
Yeah, absolutely. I mean, it's an
Yeah,
example of a reinforcement learning process. Yeah.
right. Yeah, it's and that algorithm, uh, from what I understand, is been widely recognized as the uh, algorithm functioning in the lower brain, or at least parts of the lower brain. What in these? I mean, is can you can you make that connection between what's happening, the algorithms at work in the lower brain of the human, and what's happening in these less evolved animal brains, which don't have a big cerebral cortex driving them?
Well, I would say, all of this is very much a work in progress. What I would say is there's a difference, between some of the different problems that are being addressed here, right? So with, temporal difference learning and, the kinds of things which, that style of reinforcement learning has studied, often that's about sort of figuring out what's the right set of actions, right, to maximize some reward or minimize some pain or something like that. Where you have an environment in which there's many different things you could do. What's sort of the right next thing to do, and so on. And you get feedback only once in a while, let's say, and that's sort of the big learning problem is you have to figure out what to do even though you're only getting feedback, and maybe the feedback is very vague, right?
Mhm.
About what the policy really should be, as the term is called. Whereas, with the zebrafinch, and the behavior we're studying there, it's a little bit different because you have this very precise motor activity which has to generate this complicated song, right? So the bird, in some sense, knows what it has to do. The question is, how does it set all these different parameters of very precise motor timing and muscle activations in order to reproduce that acoustic signature of the song it's trying to resemble? And that's certainly a type of reinforcement learning problem, but it's a little bit different in the sense that the it's not so much an action space; it's sort of this parameter space of all these different muscles and so forth. And so the characteristics of the learning problem become a little bit different. So, I mean, that certainly is something which human brains also have to do. We have to learn how to, walk and talk ourselves and, control our own bodies and so forth. But, I would say, so far it seems like the particular sort of style of learning that you might see, play out might be a little bit different than, something like temporal difference learning, where, you have sort of like this more higher level planning and feedback to deal with.
Yeah. Where is this going? So the NIH has come through with this money. You guys are working on it. How many labs across the country, or and are there other countries that are doing similar research? And yeah, how is this progressing? Is it progressing quickly, or is the work uh, so I don't want to say tedious, but so incremental that it's going to take a long time before you have the data on which to make conclusions?
Yeah, I mean, from my point of view, it seems to be progressing relatively quickly at this point. Um, I think up until the results from the fly brain, there was still considerable skepticism about whether this was worth doing, right? Which is a natural thing in science, right? I mean,
Mhm.
there's limited resources and people and money, and so we have to make bets about what's actually going to be useful. And I think up until we had the fly results, the community was kind of mixed. Some people thought it would be worth doing, and some people, really thought it was a waste of time. I would say since the fly brain results, there's much more consensus that there's real value in this approach, and we really want to have this data, not just for the fly brain, but for a mouse brain, and maybe one day for a human brain as well. So I think now that sort of momentum, that consensus is a little bit more in place, there's much stronger forces at work trying to propel this forward. So obviously, I just mentioned the NIH, but there's even private foundations that are now funding work in connectomics. There's other countries. The Max Planck Institute in Germany has been robustly supporting this. The Howard Hughes Medical Institute was responsible for a lot of the fly funding. The Wellcome Foundation just wrote a whole white paper analyzing the prospects for a mouse connectome. So the Wellcome Foundation is basically the largest actual nonprofit biomedical charity in the world. They commissioned basically a whole report analyzing, what it would take to do a whole mouse brain connectome. And, the thing about progress is that it's often, geometric or exponential, right? So if you go back to the Human Genome Project in the 90s, so, this was, again, a big government effort
Mhm.
in the US to sequence a whole human genome. Um, the first 7 or 8 years of the project, they achieved 2 to 3% of the whole genome. But what happened was, during that time, there was so much progress in figuring out, how to improve the methods and how to do it more efficiently, that they finished the rest of it in the last few years, right? And that's sort of the really powerful thing about these technological development projects, where you keep improving the methods at the same time as you're doing the science, and then one day all of a sudden you can do something 10 times faster than you could a few years ago, and that completely changes how people think about things. So we're sort of in the early phases of that expansion and that acceleration for brain mapping. But I would be surprised if it didn't play out. I mean, genomics is really a very high bar. They basically even exceeded Moore's law for how quickly things got better. So I don't know if we're going to do anything quite as that quite that aggressive. But even if it's a more modest rate of geometric progress, the answer would still be that within years to a decade or more, we would be in a completely different and transformative position for performing this kind of mapping to the extent we want to. That said, I think for doing a whole human brain, which is arguably the logical conclusion of all this, that's probably still some decades away. That's a millionfold
Yeah, sure.
larger than what we can do right now.
Yeah, the technology that you're working with to gather data is still this slicing, whether it's the block ablation or the
That's
salami.
right. I mean, it's some combination of slicing and electron microscopy. That said, people are really aggressively, looking at alternatives. There's, people are even using, those synchrotrons, right? Those large circular buildings, for accelerating light, and using X-rays to image tissue. There's people working on various fancy methods of using photons instead of electrons
Hmm.
to image tissue. So this is an area of biological method development that's extremely active, and there's a lot of incentive to make things better and cheaper and more efficient. And people are extremely clever in coming up with ideas to do so. So I wouldn't be surprised if 5 or 10 years from now we're acquiring our data in a substantially different way than we are
Yeah,
now.
yeah. And I was thinking of Neuralink, which manages to get these electrodes threaded through brain tissue without damaging blood vessels or with minimal damage to neurons. Where I mean, is Neuralink involved? Because there you'd be able to gather data from living brain cells without killing them in order to see them.
Well, I mean, one can debate exactly what Neuralink is or is not killing along the way, but that's a very different endeavor, I would say. So they're not really trying to map out the structure of the brain.
Yeah,
They're trying to get something into the brain so that they can, both monitor and stimulate electrical activity.
yeah,
And
but could that? I mean, is that another way that that you could gather data from the connectome?
Well, you would certainly be getting information about the brain, right? You would be getting, recordings of some neurons or some fraction of neurons in a brain. But it would be a very different type of data than mapping out the structural connectivity.
Yeah.
There are other methods for recording activity. So people, for example, have invented something called a light sheet microscope, where you can put a whole baby fish into it and image all of the. You can take a movie basically that describes the activity of every cell in that brain
Hmm.
over time. And those are very exciting data sets. But scaling that to a human is
Yeah.
extremely improbable at this point, at least.
And what's the end ambition that eventually you would map this mouse brain, you'd be able to recreate it with artificial neurons in a computer, uh, with programming, uh, and then run different algorithms to see. I mean, yeah. I mean that in my mind, that's where this would go. Is you have this computer program that is basically an analog of the mouse brain, and then you're playing with different algorithms to see how the thing should be working or could work. Is or am I in science fiction land?
Well, I don't think you're in science fiction land. I mean, I think, look, I mean, ultimately the goal is to understand how the brain works, right?
Mhm.
I mean, that's why people fund this type of work. I mean, um, that's really the first order of business. And more specifically, the reason people want to understand how the brain works is to improve human health, right?
Right.
I mean, that's why the NIH funds this work. That's why HHMI and Max Planck fund this work. Ultimately, the goal of all of this is to improve medicine, which is quite appropriate given the public investments
Mhm.
in the space. But in order to do that, we need to figure out how the system works and what goes wrong when it doesn't. And for the brain, that's enormously difficult, right? If you think about the kinds of treatments that we have for, mental diseases or neurodegenerative diseases and so on, it's quite abysmal, frankly. Like not a lot has changed over the past few decades. The drugs that we have to treat, very common mental health problems or more complex ones like Parkinson's and so on are not great. And there's really an enormous need, right, for
Mhm.
better options. So the main goal of all of this is to aid in that endeavor. But that's super high level. I think once you get a level below that, the question is, what are the different tactics that you might take in order to make progress on that question? And indeed, one approach might be, okay, let's have computational systems that can reproduce aspects of the brain so that we can study the brain more efficiently.
Mhm.
If you think about, something like what's happened with proteins recently, where we can computationally go from a sequence of amino acids to the 3D structure, that dramatically accelerates the kinds of things you can explore and the number of options you can, investigate. Similarly, if we had, computational models of the brain or parts of the brain that were, realistic, we can much more quickly explore, different options for manipulating the nervous system to treat some of these issues, or figuring out what the basic parameters and principles are. So I would say those are the main goals, and we have a lot of work to do before we sort of realize them.
Yeah, but wouldn't there also be, wouldn't this inform artificial intelligence research? Uh, I mean, there could be architectures or, um, as you say, I mean, the brain operates on very low wattage and very little data compared to, uh, a large language model, for example. Uh, couldn't this wouldn't there be insights to building uh, AI models that could perform in ways similar to the brain?
So it's possible. It's just a question of who makes faster progress in some ways, right? And as far as I could tell, my colleagues on the AI side of things are um, things are moving along very quickly there, and the question is, to what extent do they really need insights from biological systems to keep making progress? And there's no, it's hard to answer that question
Yeah,
definitively, right? People have different intuitions, and certainly if you think about how they got to the present day, where did these systems come from? A lot of that came from folks like Jeff Hinton, paying attention to basic principles about how the brain works, right? So this idea that, artificial intelligence systems should be made out of deep learning systems, right? That's not an obvious choice to make. And in some sense, for a long time, people had other ideas, but, it was folks like Jeff Hinton who were inspired by very basic aspects of brain organization that kept pushing on that approach. And that's really what got us to where we are today. Now, as to whether the next 20 years of progress will need some additional insight from biology, it's hard to know. If they're going to run into some sort of wall and run out of ideas, then maybe, or maybe at this point, they're just going to keep engineering those systems to be better and better and more efficient. And maybe at that point, biology is kind of irrelevant. Who knows?
Hi, this episode is sponsored by Celonis, the global leader in process mining. AI has landed, and enterprises are adapting, giving customers slick experiences and the technology to deliver. The road feels long, but you're closer than you think. You see, your business processes run through many systems, creating data at every step. Celonis reconstructs this data to generate process intelligence, a common business language. With process intelligence, AI knows how your business flows across every department, every system, and every process. With AI solutions powered by Celonis, enterprises get faster, more accurate insights, a new level of automation, and a step change in productivity, performance, and customer satisfaction. Process intelligence is the missing piece in the AI enabled tech stack. Search Celonis, Celonis, to find out more. That's it for this episode. I want to thank Viren for his time. If you want to read a transcript of this conversation, you can find one, as always, on our website, IonAI, that's EYE-ON.AI. And remember, the singularity may not be near, but AI is changing our world. So pay attention.